Coimbatore Local Center under the aegis of Chemical Engineering Division Board of IEI in association with Division of Biotechnology, Karunya Institute of Technology and Sciences, Coimbatore. You know, the IEI is a very prestigious professional body comprising over 2.6 lakh engineers from various disciplines. Headquartered in Kolkata, IEI operates through 124 centers across India and 6 overseas locations. Our Coimbatore Local Center has been actively contributing to the engineering community for the past 60 years. At this juncture, I am pleased to share that IEI headquarters has presented the Best Local Center Award to the Coimbatore Local Center for the year 2024-25, recognizing its overall performance among 90 plus centers nationally. IEI supports a wide array of technical activities, including guest lectures, workshops, seminars, webinars, national conventions, statutory day events, and things like that. So today's program is one such initiative. IEA also provides R&D funding for diploma, undergraduate, postgraduate, and full-time PhD scholars. So I request those who are pursuing the degree program can approach IEA for R&D funding. So as the chairman of the IEA Coimbatore Local Center, I am delighted to deliver the welcome address for this technical webinar. I welcome the president of IEA, chairman of Chemical Engineering Division Board, members of Chemical Engineering Division Board, council members, and Saurav Dasgupta, executive IEA headquarters to this webinar. I would like to extend a heartfelt welcome to our distinguished panelist, Dr. Vivek Suresh Dalvi, Program Manager, IIT Bombay Research Cup for Green Energy and Sustainability, Bombay, Dr. K.K. Prashant, Director and CEO, Symbion Labs and Scientific Services Private Limited, Chennai, and Dr. T.S. Manihanda Maharaj, Co-Founder and Director R&D, Xenobiomic Research and Technological Development Private Limited, Puducherry. Of course, they will be sharing their expertise on the green biotechnology for environmental sustainability. I also wish to welcome Dr. A.K. Priya, Committee Member, IEA Coimbatore Local Center, and Professor, Division of Biotech, Karunya Institute of Technology and Sciences, who have taken the responsibility as moderator of this event. At this juncture, I would like to express my sincere gratitude to the Dean, Engineering and Management of KIT for their continuous support and collaboration with IEA Coimbatore Local Center in organizing various events. Additionally, I would like to welcome Dr. J. Pradeep Kumar, Honorary Secretary of IEA Coimbatore. I am pleased to welcome the committee members, IEA corporate members, faculty, staff, and students from various institutions, as well as participants from various sectors across the country for this program. This webinar highlights the growing importance of sustainable solutions to address pressing environmental challenges, calling for innovative and eco-friendly approaches through green biotechnology. The theme of this webinar brings together key domains that are shaping the future of environmental sustainability. Green biotechnology is emerging as a powerful tool in areas such as bioremediation, waste management, and the development of bio-based products, significantly reducing environmental pollution and promoting resource efficiency. I am sure this program will provide a valuable platform for IEA members, experts, researchers, and professionals to share their insights and explore innovative approaches toward achieving environmental sustainability through biotechnology. So to all participants, I wish an insightful and enriching experience during this webinar. On behalf of the IEA Coimbatore Local Center, I once again extend my warm welcome to all of you. Thank you. Over to Dr. Priya.
Thank you, sir. I'm happy to welcome today's first guest. Dr. Vivek Suresh is an accomplished researcher and innovation leader in the field of environmental biotechnology and sustainable engineering. He completed his PhD from the IIT Delhi, where his research focused on developing advanced photobioreactor system for wastewater treatment and biomass valorization. Currently, he serves as a program manager at the Green Energy and Sustainability Hub, IIT Bombay, where he leads multidisciplinary initiatives in urban sustainability, carbon capture, and green energy transitions. His work bridges academia, industry, and policy, driving impactful solutions for climate resilience and sustainable development. Dr. Vivek research expertise spans circular economy approaches, carbon dioxide removal technologies, life cycle and techno-economical assessment, and microalgae-based carbon capture systems. He has successfully led a major BIRAC-funded project on wastewater-derived microalgae supercapacitors, contributing to carbon-neutral biorefinery concepts. With over 400 citations and multiple high-impact publications, patent, and international conference presentations, he has made significant contributions to advancing algae biotechnology and environmental sustainability. His work has been recognized through prestigious awards, including the Biotechnology Innovation Grant and Research Excellence Honors from IIT Delhi. Today, he will be sharing his insights on cutting-edge innovations in sustainable technologies and environmental biotech.
Where we start?
Over to you, sir.
Thank you, Dr. Priya, for a very warm welcome and introduction. So I will share my screen.
Yes, sir.
So my screen is visible, right?
Yes, sir.
Okay. Thank you. Thank you, Dr. Priya, once again. Hello, everyone. I'm Dr. Vivek Dalvi. I'm a program manager for research activities at Green Energy and Sustainability Hub, which is at IIT Bombay. Uh, I will begin with my presentation. So my presentation title is The Role of Microalgae in Wastewater Treatment and Resource Recovery. We would like to understand that what is the global wastewater challenge actually? When we talk about the wastewater, so it is not an individual problem, it is a problem of the society and communities as the wastewater generated is being directly disposed of the environment without the treatment. That causes a significant harm to the environment, to the life below the water, and also to the surrounding people who are living next to these water channels which are going directly to the rivers uh, or the sea. So this generation challenge is majorly accelerated due to the highly population growth and industrial expansion. And the conventional wastewater treatment technologies such as activated sludge process or similar to that bacterial processes face significant limitation because of they either would require very high energy requirements for the operations. Operations are generally non-stable and substantial growth of the sludge would happen, and that would lead to elevated operational cost and failure of the most wastewater treatment facilities. And that is why there is the urgent need for very sustainable low energy uh, solutions are required, for which in our understanding microalgae based systems are very promising alternatives that would change the scenario, have the potential to change the scenario of the wastewater treatment, and parallelly will minimize the environmental impact associated. So, first, we would like to understand what are micro or macroalgae, which are being used nowadays for the wastewater treatment, or the research is majorly ongoing. So, microalgae are nothing but very microscopic photosynthetic organisms that simply converts the sunlight into the chemical form through the process called as photosynthesis, as normal conventional any normal plants will do. But these microalgal systems are nothing but at least 10 times higher in process with respect to conventional normal plants are. And also they are ubiquitously very uh, abundantly present in nature with respect to multiple freshwater lakes, marine systems, or also wastewater side streams are also a major source of different kinds of microalgae. And since it is having very high, also it is having very high and fast reproduction rate, and exceptional nutrient uptake capability. That makes very unique in order to have the ideal candidate for the wastewater treatment. The common genres for of the microalgae which are being conventionally used in wastewater are such as multiple chlorella species or syndesmus species are there. Also, at some times, species like spirulina are also being used for the wastewater treatment and uh, yeah. So the role of microalgae lies does not only in the wastewater treatment alone, but along with the wastewater treatment, it generates a valuable biomass along with it. So that is the additional advantage. Uh, so why microalgae are exceptional for the wastewater treatment are nothing but it is having efficient nutrient uptake capacity, such as through the process called as photosynthesis, it effectively utilizes the nutrients such as nitrogen and phosphorus, which are required for their own growth. And parallelly, it also helps in removal of the heavy metals, pharmaceuticals, and chemical compounds in trace elements. That makes it a very unique in nature. Also, it has parallel application in the carbon sequestration, such as it converts CO2 captured from the airstream or the dissolved in the water for the as a carbon storage within the biomass, and which also contribute to the climate change and mitigation parallelly. Uh, Third advantage is that the minimum sludge generation, like in any conventional process, the algal biomass generated has minimum residual sludge, and it has certain third point of application, such as in the form of biofuel or the animal feedstock, or in any other third format. So which reduces the disposal cost and environmental burden um, for the waste water treatment. And simultaneously, as this the biomass is happening, so it gives the economical opportunities from the generated from the waste streams. So that is the unique advantage of the position of this microalgae wastewater treatment technology compared to conventional processes. So as I just told you, what is the mechanism of pollutants removal? So like for conventional wastewater treatment units, the carbon, nitrogen, and phosphorus are the primary contaminants present in the wastewater that will increase the BOD or the COD of the wastewater. So, in this case, microalgae assimilates these nitrogen phosphorus for their cellular biomass growth and metabolic activities. Along with that, as I told you, that heavy metal biosorption will happen through uh, its surface, through the surface binding and intracellular accumulation, such as highly toxic metals like cadmium and lead also. But it is not at relatively very high concentration, at low concentrations only. If the concentration of heavy metals is very high, let us say in milligrams per liter, so then that will act negatively to these living microorganisms, usually kill the microalgal or bacterial cells and will not lead to the heavy metal removal. But at relatively lower concentrations, which are present in the municipal wastewaters, this will work fantastically. So uh, it helps in the degradation of the organic matter, since it is uh, not only algal uh, uh, not only algal system alone that works in the wastewater treatment. It is a combination of the synergy of algae and bacteria that helps the breakdown of the complex organic compounds into the simpler form, so thereby removing the ultimately the organic matter or the chemical oxygen demand present in the wastewater. And the very most important is that it also helps in the reduction of the pathogens from the wastewater. So there are multiple technologies such as UV irradiation or the ozonation, that is nothing but the oxygenation that affects the create the hostile conditions for harmful organisms for pathogen removal. But in particular to that, since it is a photosynthetic process, so with the help of the sunlight and microalgae, oxygen production from the ROS produced within the wastewater will ultimately help removal of the pathogen reduction. Also, there are multiple physical parameters such as pH, temperature, that also play key important role in the parallel removal of the pathogen reduction. So, while talking about microalgae and microbacterial conservation, uh, so algae and bacteria are self-sustaining ecosystem for the wastewater treatment. While as algae produces oxygen through photosynthesis, which supports the aerobic bacterial metabolism. As these aerobic bacteria will consume this oxygen and eventually produce the carbon dioxide in the form of dissolved bicarbonates, which will enhance the microalgal growth. In that way, the what researchers generally observe that there is the enhanced removal of chemical oxygen demand through this combined action. Rather, we go standalone for the only bacterial systems or very pure algal systems. So, algal and bacterial mixed consortium systems generally perform. Uh, if we go standalone for the, let us say, bacterial wastewater treatment system, we would need high amount of aeration in order to support the bacterial growth in the as oxygen is required. But when we couple up the algae and bacteria together, the aeration would not be required. So ultimately, the cost for the aeration goes down, and that has the better uh, advantage. So there are multiple types of wastewater treatment systems, such as there are open pond systems, high rate algae ponds, photobioreactor, and biofilm based systems. So open raceway ponds are nothing but very simple, low cost algae cultivation systems where mixing is happened with the help of only paddle wheels. Compared to when we look at we look for the very high like a little better performance efficiency, then people go towards the high rate algal ponds, where the mechanical mixing is being achieved very properly compared to conventional plant. Depth of the algae cultivation system is being optimized. The paddle wheel efficiency also, the carbon supply will be through the CO2 source will be given to these high rate algal ponds. So the energy is defined in a way that the means are provided for these algal ponds in a way that there is a high rate of algal production. And then comes we can come to the third one is which is nothing but the photobioreactors. So photobioreactors are nothing but the closed systems which offer very high productivity of the biomass. It also offers the less contamination with respect to any contamination from the air or the water, and it also supports the year-round operations as temperature, pH conditions can also be very well maintained. And there comes the last for the LD cultivation, which is significantly having an advantage over um, other systems in the terms of harvesting of the biomass. Because in the biofilm systems, the microalgae grows along with the bacteria on the attachment platforms, and we just simply need to scrap the grown biomass after some time period in order to harvest the biomass for any third uh, third application of the resources recovery. So when we look at the photovarieties in detail, so the photovarieties are actually the advanced cultivation systems, as we can see as shown in the figure, which gives the controlled environment for the microalgae growth. So it is having these are in different forms. Like as we can see in this image, is nothing but the tubular photobioreactor. Likewise, there are photobioreactors which are called as flat pattern photobioreactors, where the surface is very flat. Also, there are recirculation photobioreactor systems as well. So the advantage where we can achieve is that the high biomass growth, the lower land footprint would be required, the optimized light distribution and CO2 utilization can be happened. So the protection of the microalgae from the external environment predators can be happen. and year-long operation of the photovoltaics can be achieved despite it is very low temperature conditions or very high temperature conditions, or uh, monsoon season is there or not. So it gives a better control over the system. Uh, when we look at these microgrid irrigation systems, let it be all of any type, there are certain key operational parameters to achieve good health of the microalgae culture within the ponds. So first parameter is the light intensity and photo period. So the amount of light um, is plays a very key role, as low light conditions or very high light conditions are not really required. So the when we look at the geographics of the countries, like let us say in European or extreme south countries, so those conditions are not very ideal for the microalgal to achieve the highest efficiency of microalgae growth. So certainly there needs the optimization. Also the pH management. So optimal range for the pH is around 7 to 9, and also, if that range goes beyond, then it affects the nutrient availability and the microbial metabolism. So there are multiple strategies through which pH can be managed, such as uh, acid alkali supplementation periodically, or the provision of CO2 supply in order to make the culture from more alkaline to acidic in nature. uh, yeah, so in that capacity. The third parameter is the mixing of the culture media. So how it can happen. So conventionally, there are two types. Mixing can be happen through the mechanical mixing, and the second option comes is the mixing through the aeration or the CO2 supply. So both of these means are there, uh, and their ultimate goal is to provide the uniform distribution of the microalgae within the culture media in order to provide them the better exposure of the sunlight. Then comes the very important point, such as temperature, where like temperature 30 degrees Celsius is required for ideally it is required for uh, microalgae growth, and there are multiple species which require which has multiple temperature ranges, such as extremophile microalgae are there, or low temperature microalgae systems are also there. So it is depending upon, again, species to species, but ideal temperature range lies between 20 to 30 or 35 degrees Celsius for the better optimal growth. Uh, then the hydraulic retention time. So the contact time between wastewater or the nutrients present in the wastewater, uh, 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 along with to the microalgae, is the very important thing. So generally, the retention time is kept very high in order to have the better microalgae growth. But also, we need to take account of the both, like what is the HRT of the wastewater treatment system and the HRT of the microalgae growth. And finally, the CO2 source supply. So as much as the carbon supply will be there in the form of inorganic carbon, so better will be the growth of the micro. So conventionally or generally, the CO2 supply is given through either through in the form of um, flue gases, or the CO2 directly from the biogas operating plants, or such that. So if we could control these all six parameters, so then the microalgae photobioreactors or the yeah, and finally looking at the resources, and having a very good resource recovery, whatever the microalgae biomass we generate after the wastewater treatment, having the applications for the biofuels, primarily for biogas through hybridization, or also it could be fermented to the bioethanol production through the carbohydrate fermentation. Also, there are multiple applications, such as the production of bioplastic materials such as PHAs or PHs, which after extraction of these PH or PHB, the biopolymers could be formed, which has the application into the packaging materials. Also, if the biomass generated is rich in proteins, then it could be act as a animal feed source. But depending upon the like how much cross contamination within these biomasses with respect to any heavy metals or something like that is there or not, we need to check, and depending upon that only further explore. But but from the looking at the high value product point of view, the pigments what we generate, there are certain species of the microalgae such as astaxanthin, for the production of astaxanthin and beta carotene. So these kind of pigments can be parallelly cross harvested and can be having better and which has a better market or the specialty chemical values are there. So these could enhance the viability of the wastewater microbial potential. So, as I told you that there is the very big potential of the through the point from the point of view of carbon dioxide potential. So, as like there is like theoretical potential of CO2 per meter square per day capacity is there. So that can help in the climate change mitigation. Also, if it can be integrated with flue gas. Industries and carbon negative systems can be achieved. Uh, like a lot of research is happening, and many industrial scale processes have already been developed and piloting, and there is a research happening at the very good at the pilot scale. Amount. Yeah, so at what kind of waters which we can target primarily upon, so we can segregate the wastewaters into multiple formats, such as municipal wastewaters, which are nothing but having the low nutrient, low heavy metal contamination, but having the significant amount of pathogens into it. So this kind of wastewater is that industrial effluents. We cannot directly go for the industries such as heavy metal industry or mechanical industries with these kind of system, but we can certainly target with the kind of industries which are producing the organics or the something like that, such as food processing industries, beverages, or the textile industries. And very good algal material systems also show very good performance with respect to agricultural runoffs, as agricultural runoff water has a lot of fertilizers into it, so that helps the microalgae to grow in a better way. Also, that prevents the eutrophication of the potential water bodies where the agricultural water goes off. And certainly, many decentralized systems have been already been designed at many places at small and moderate scale. So that depends upon how one utilizes that emission. So after having all this discussion on how microalgae grows and what are their parameters to grow and cultivation, we need to check on the what are the advantages and limitations of these systems are. So key advantages what we see is through from the point of view of energy efficiency. In a way, if we could go for the algal bacterial systems, also we get the dual output such as uh, biomass for the biofuels, feeds, fertilizers. Also, since it is a chemical free, we don't need any coagulation processes or something like that. That lowers the cost, and also it can achieve the circular economy approach as well. We need to also accept that there are certain challenges, such as a very high footprint compared to what conventional wastewater treatment facilities have. So that goes 2 to 3x of the land requirement. Also, the cost for the harvesting, since these are distributed in the uniformly distributed water, so harvesting takes time, and it would need again the chemicals or the coagulants for the any like open raceway or photobiotic system, but that will not the case in biofilm systems. Also, the variability in light and temperature affects the year-round performance. Also, there are scale-up complexities such as like how like systems up to the pilot seems feasible, but at very large scale, how to handle the large amount of biomass produced. And again, it's a disposal of the third like it's a resource recovery application. These are the few pointers which are into the current challenges. Yeah, yeah. From with this, I would like to close my presentation, and I would like to ask anyone if they want to have any questions for me in order to answer very specifically. Yeah. So I am open for the any questions.
Yes, sir. Thank you so much for your session. And a few questions were asked by the participants that what are the major challenges while scaling up this process?
Yeah, yeah. So the major challenge is that primarily when we take account of scaling it up from, let us say, research lab to pilot scale and pilot to commercial scale. So first thing from the point of view of wastewater treatment, the challenge is with respect to uniform mixing of the liquid medium or the microbial medium. For let us say for a raceway pond kind of system, we would go for at least 1 acre area of the cultivation system. But then for that, how to optimize the paddle wheel speed, the ratio, the contact between the water and the paddle wheel support, all things are need to be taken into consideration. The second challenge is with respect to the contamination. The contamination is generally prone to happen with open microalgae cultivation systems, but that is not the case with the closed photovoltaics, where we have better control over the environmental conditions. But again, when we say that there is the better control with respect to closed photovoltaics, these photovoltaic systems are relatively expensive, and then that affects the scalability criteria, uh, scalability thing.
Okay, next one is like another question is syncoms, whether the syncoms enhances the algal wastewater treatment performance, like synthetic
What? Uh,
microbial communities. Synthetic microbial communities, will it enhances the algal treatment?
Synthetic microbial communities. Okay.
Yes.
We can try for the synthetic consortiums like combination of different algal bacterial uh, species would be beneficial for the wastewater treatment. But ultimately, the species of the local microbial or bacterial species will dominate over a period of time. Okay, so that may would happen. But if you want to like maintain your own species of own species of desire, you would need to periodically have to keep on supplementing the some seed culture of the species, your desired species. That may help it overcome the problem of domination of um, the native algal or bacterial species over time.
Next question is like, are you coupling the microalgae system with any AIML optimization or something like that?
Yeah, people do the AML combinations or the studies with respect to herbal bacterial wastewater treatments, where they simulate different environmental and process parameters altogether with respect to literature available, and then that cross validation can be done. I personally don't do it, uh, but uh, people who are having expertise very well in the AML domain, they do generally. There is a lot of literature available on the same. Yeah.
And the last question is, usually if we think about the microalgae, we usually think about the cost effectiveness. How far
Yes.
it is very cost effective compared to other treatment technologies?
Yeah. Yeah. So with respect to algal waste, like it is comparatively expensive when it comes to the cost, because algal systems have a lower aerial footprint, so ultimately the land requirement would be higher. Uh, so that is the main first thing for the practice like wastewater treatment, where the end comparatively when we look at the algae for the nutraceutical production or something like that. So having the combination of something like microalgae for wastewater treatment as well. But the end biomass generated should have um, some value associated with it. So that may lower down the economic requirements or the economic benefit.
Thank you, sir. Thank you for the valuable session which is given by you for this webinar. On behalf of Karunya Institute as well as IEA, we thank you for your presence over years. So we'll
Yeah,
move.
yeah. I'm also grateful towards the organizing committee, the Karun University, the IEI chapter, for giving the opportunity. And yeah, it was pleasure.
Thank you, sir. And we will move on to
Thank
the
you.
next session, next speaker. I welcome Mr. Prashant, is a distinguished bioengineering professional with over a decade of experience in drug discovery, testing, and applied life science. He holds PhD in bioengineering from Anna University and has built a strong reputation as an entrepreneur, researcher, and academic contributor. He is a founder, director, and CEO of Symbion Labs and Scientific Service Private Limited, and also serves as a co-founder and technical head of Fine Biosolutions, focusing on bio-based products and sustainable solutions. Additionally, he is actively involved in agri-tech innovation through the Farmfinity, uh, where he lead product development. Mr. Prashant has extensive academic engagement as an adjunct faculty in biotechnology and serves on multiple boards of studies and consultancy committee across reputed institutions in Chennai. As a mentor and guide, he has supervised numerous PhD scholars and over 85 UG and PG research projects as well. A passionate knowledge disseminator, he has delivered more than 200 training programs and workshops, along with seminars and conferences at national and international levels. His research contributions include several international publications, and he has actively participated in and co-chaired technical forums and panel discussions. Today, he brings his expertise to share insight on cutting-edge development in biotechnology innovation. With this intro, I welcome you, sir. The session is over to you.
Yeah. Am I audible? Yeah. Okay. Thank you,
Yes,
ma'am.
sir.
Yeah, ma'am. Thank you. Thank you so much. Thank you for the wonderful introduction and thank you for the opportunity. From Karunya Institute of Science and Technology and Science, as well as Institute of Engineers. So, as the warm introduction has given to us, so I will directly jump on to the session and yeah. So, myself, Dr. Prashant, CEO, founder, Symbion Labs. We are a preclinical drug discovery laboratory. So the idea is to put up a word in terms of how well we can use alternate animal model in terms of green chemistry. So utilization of different models, especially alternate animal model, which serves as a wonderful tool in terms of identifying the toxicity and efficacy aspects of products from green technology like nanoparticle and fertilizers, biopesticides, and utilization of this model to enhance the toxicity, efficacy, as well as to increase the robust efficiency of green technology outputs and products. So, myself, we are running a preclinical drug discovery firm, and we are having alternate animal models. Apart from the conventional model that we are using in our day-to-day life, we are using alternate animal models like zebrafish and yeah, like zebrafish, Drosophila, and Artemia, and brine shrimp, and other different animal model which can be utilized to increase the efficiency of high throughput production, especially production of greener and better materials, and as well as utilization of lesser utilization of conventional animal models like rat, mice, and rabbit, whether we can use our model instead of this conventional traditional model. So this is what we are doing as an organization. And this is the ideology of today's webinar. So the green product outputs, which includes phytochemical and plant extracts, biopesticides, biofertilizers, microbial metabolites, and novel biological products, requires a different approach for its toxicity output. So putting it in a cell line, putting it in a rat model, and spending traditional monetary requirements to produce alternate or a greener product is not the sustainable output, right? So we are using green technology to increase sustainable output, as well as we are using green technology to put better economical sustainable products onto the market. But utilizing the traditional models like a rat or a mice or other conventional models to study its toxicity and efficacy is not only a waste of time or economical resources, but also it is not the apt or sufficient model towards the green products. So the challenge in terms of having a traditional model with respect to our green products, the major challenge is that the environmental impact. So ecological impact is there. Putting lot of mice or rat or rabbit in a cage to study a cosmetic product, to study a food product is not sufficient sustainable output, right? So we are providing an alternate solution. We are trying to create awareness about an alternate solution, whether we can use zebrafish, whether we can use the embryo of zebrafish, whether we can use a drosophila, whether we can use artemia, whether we can use in silico AI models which are available in the market to put something called replace, reduce, refine. The 3R or the 5R strategy, which revolves around utilization of alternate animal model instead of traditional animal model, especially in this particular aspect towards the studying of greener products. So to put it into retro perspective, generally we as a preclinical firm, we go with the traditional drug discovery route. If you take up thousands of compounds or hundreds of compounds to put it into market, it takes $1 billion to put a product onto the market. So if every time we have to spend $1 billion to put a product onto the market, the greener sustainable output, especially towards the economical resources, is not validated. So instead of utilizing traditional animal model, if we can able to use an in vitro model or an in vivo model, an alternate animal model, it reduces and saves up to 25% of the economical resources we are putting onto drug discovery or product development. So traditionally, we will be using traditionally we will be using different approaches, in the sense, if you have a drug or if you have a chemical entity, if you have a molecule, we'll be trying to repositioning is to reduce the cost effectiveness. For an example, there would be a pre-existing drug for a particular ailment or a pharmacological requirement, which nowadays people are taking it and using for some other remedies for some other issues. Yeah, I will come back to the questions. Yes. So moving ahead. So 1 second. Right. So if you talk about alternate animal models, how reliable are them? Can we utilize this model for any purposes? So what are the purposes for any particular model? To put it into retro perspective, there are so many traditional models. Like you take a rat or a mice, you can put it in a cage. You talk about the failure of utilizing conventional model. For an example, every time you take up going for a traditional model, you have to go for your CCSCA committee or CPCSCA committee. An animal ethics committee is there. There is no harm and foul, and we have to definitely follow and strict adherent towards the ethical requirements. But going further, you have 10 different combinations or 50 different combination of a small molecule, or like a drug molecule, or like a plant extract. Every time you cannot go and ask for 10 animals to 50 animals. So taking up a conventional model means putting up larger amount of money. Alternate animal model where it can be utilized. Each model has its own pros, each model has its own cons. For an example, certain alternate models can be utilized for particular applications. These models are the common alternate animal model that are currently available in the market, which can be utilized for greener products. For an example, initially we'll start with in silico computational simulation. So you talk about in silico computational simulation. There are more than 50 to 100 different pipelines which are available in the market in terms of utilizing in silico compound for in silico approach for estimating efficacy, bioactivity, and toxicity of a compound. For an example, you take a structure of the compound. With the structure of the compound alone, we can able to predict its toxicity. We can able to predict its bioefficacy. That is called as QSAR, quantitative structure activity relationship. With mere 2D structure itself, we can able to predict the 3D structure. From the 3D structure, we can able to predict the environmental impacts or alerts. So that is how we can utilize any particular 3D or 2D structure without directly synthesizing a compound. We can able to understand its toxicity. So these will be useful towards, for an example, developing a nanoparticle or a nano solution. Directly before going on to develop a plant extract along with a nanoparticle, we can take the list of plant compounds. We can take in the list of plant compounds which are available. So go to a GCMS analysis or a LCMS analysis, take a list of plant compounds, put it in a biological active AI predicting tool like SwissADME is there, admit property prediction is there. Especially towards toxicology, there is a tool called EPA. EPA's tool called EPI, Environmental Protection Agency of US, has released a tool called Environmental Protection Institute Software. EPI suite is there. You take up this EPI suite, you put your 2D structure of your chemical compound, you put the 2D structure of your gaseous compound, it will give you the outputs. What kind of output, whether it is causing toxicity to a land mass, whether it is causing toxicity to a water mass, whether it is causing toxicity to a gaseous mass. Different environmental models are there for different toxicants, which is introduced and released by EPA, Environmental Protection Agency of US FDA, Food and Agricultural Association. Food Drug Discovery and Agriculture is also there. So you take up in silico, which I'm not going to take too much time. The in silico aspect is that with merely a structure, you can able to predict toxicology of a compound. With merely a structure, you can able to predict bioactivity of a compound. With merely a structure, you can able to predict how the compound is going to interact with the protein. Something called docking is there, something called biosimulation is there, which we are using in our day-to-day drug discovery pipeline in our laboratory, where we will draw a structure of a compound, put it along with the protein, simulate for 100 nanoseconds. During this 100 nanoseconds of time, the protein structure will interact with your larger molecule. The protein structure will interact with your larger molecule, and it will give you an output which tells the protein is going to get interacted with small molecule. This bioactivity is there. So how well we can introduce this particular tool for green chemistry is that taking up the list of biological entity which are already available in the market, a pollutant which is already available in the market, and identifying is the bioefficacy level without directly going on to your wet lab experiment. Using a regressional analysis or using a network analysis, you can able to predict the particular toxicity of the compound. So these are some of the basic tools which are available. So this is the gist of what else you can do with bioinformatics. But moving ahead, bioinformatics is very basic prediction level tool, which tells you with the structure where you can able to predict a compound or not. With the prediction itself, where you can able to say where I'm going to synthesize this compound, this much of entities, if it is added, if it is going to be toxic or not. But real-time toxicity evaluation, we can use cell culture models. Organoids are there. Bio-based, like a human cellular-based models are there, which is already called as organoids or like in vitro cell techniques. So for an example, I take up a cell line, a human cell line or a rat cell line, I put it in a Petri plate, I put my toxic chemical like a nanoparticle-based biopesticides. I can able to understand what kind of bioactivity or what kind of toxicity it will give to the reduced required toxicity level, right? So this model will be the beginning of predicting toxicology for greener products like nanoparticles or pesticides. But the apt model, one particular apt model which you can directly take in not only the basic toxicity but also product development is zebrafish. So today's entire session that we have prepared about is that go ahead to with zebrafish. Why and how we can use alternate model, especially zebrafish, in terms of developing a greener product. So why zebrafish? So why and how zebrafish? To talk about initially zebrafish, it is an animal which is originated from. It is an animal which is originated from the banks of river Ganga and as well as river Brahmaputra. So zebrafish is a common animal which is available in the Indian subcontinent for longer period of time. Zebrafish has been utilized in terms of drug discovery for more than 4 to 5 decades. From 1970, 1980 itself, zebrafish has been utilized in terms of drug discovery. So many toxicology studies has been done with zebrafish. But when you come back and trace to the origins of zebrafish, it is a model from Indian subcontinent. So it is not like an alien model from a different continent, or it is not a model which is not available in India. It is a very common model which can be utilized, I mean, easily procured in India itself. You talk about zebrafish. There are different types of zebrafish available in the market. The common zebrafish you take up from the market will be like tetra, neon in color, or like green in color, or like vibrant colors. Those are nothing but your ornamental fishes which are for domestication purpose. You can grow it in an aquarium, or like you can grow it in your home. But the strain of zebrafish that we are talking about is called as wild strain. The wild strain of zebrafish will be like either blue in color or black in color. The stripes. Okay. The zebrafish, then from the name itself, the common analogy is that zebrafish is a fish, a small 2-inch animal, which will be having strides in its body. And the strides are either blue or black in color. If it is blue in color, we will be calling it as AB type strain. If it is black in color, we'll be calling it as W2 strain. So these are the two strains, common strains, which will be utilized for scientific purposes. And where we can get these strains? These strains are also available in the market. A common aquarium shop can also be available, or else you can even procure from the Central Research Laboratory. STIBA is there in Chennai, where we can procure it. In terms of when you take and talk about South India, there are certain institutions like IISCR, Trivandrum, they are working on zebrafish. So breeders, okay, these breeders are like CCSC approved breeders where you can buy your zebrafish. So, okay, so we can buy the zebrafish. How we can utilize this zebrafish for greener technology solutions? Yes. So moving ahead, the best part about utilizing zebrafish as a toxicology animal is that breeding is very, very, very easy. You can take in a pair of animal, you can take in a male and a female. If you can see over the screen, okay, the male will be like a little darker in color, brighter in color, little vibrant color strains would be there, whereas the female will be like having a bulge abdomen. For an example, the A is male, whereas B and C are female over here. So the male zebrafish will be darker in color, and the female zebrafish will be having a bulged body. The bulged body is nothing but female zebrafish used to carry about minimum of 300 eggs. So 300 eggs will be there at a single stitch. The advantage is that when you take up a male, when you take up a female, you put it in a breeding tank. You put it in a breeding tank. Okay, you allow it grow. I mean, you allow it for breeding for some period of time. During the first ray of sunshine, like early morning, the breeding will occur. Generally, zebrafish is an animal which will mate very specific towards the required animal. For an example, there is not group mating. Their group mating is also available, but in terms of scientific experimentation, we will take one pair, like one female, one male, we'll put it in a single separate breeding tank. And after some time, during the early morning or later evening, you will get the embryo. Why it is that? This is an ex situ fertilization. Like some all other fishes, zebrafish is having an ex situ fertilization. And the major advantage is that this has a transparent embryo. Optically transparent embryo you can easily collect. An optically transparent embryo you can easily collect. You can put it in a simple optical microscope. Even through naked eyes also you can able to see. So even through naked eyes it will be like small bubble. If the bubble is transparent means fertilization is occurred. Okay, if the ball or bubble is not transparent, it is opaque means pearl in color means transparency is not there means fertilization is not there. So transparent embryo means fertilization is occurred. Transparent embryo means fish is healthy. I mean the embryo developing fetus is healthy. So the development phase occurs. The development phase occurs within the first 72 hours of the fertilization. So this is where we are going to use zebrafish embryo for toxicity study, especially for pesticides and fertilizers. You think about your greener products. I am developing a pesticide or a fertilizer or a microalgae feed, or like any particular product that I am going to put it in the environment. If it is going to release onto the environment one way or another, it will definitely reaches into your waters or rivers or streams, right? So if you are putting a fertilizer, you are putting a pesticide, one way or another through your irrigation canals, it will reach the water streams like a pond, like a lake, like a river. Okay. Higher concentration of these compounds in each particular model, like a water model, like a pond or like a river, either create algal bloom, eutrophication is there, or it will create some other toxic and problem towards the normal ecobiosystem, right? So what we need a fish model to study the pesticide. We need a fish model to study the fertilizer. And you take up a zebrafish, you take up the embryo of the zebrafish. The first 72 hours of the embryo, as you can see in the picture, you can easily map its morphogenesis. So the morphological features is nothing but the direct visual cues which you can predict and map towards the actual toxicology efficacy of your pesticide or a biofertilizer. So you take up a pair of zebrafish, you put it in a tank, you wait for the morning. Early morning, you can get 300 eggs. You can collect the eggs, you can put it in a microscope. You will be observing this. This is the developmental growth phase of a zebrafish mounted onto a normal microscope. The video is taken from Harvard Medical School. So it's a valid output where you can see the animal development through its normal microscope. Here you can able to observe there are two different poles. One is vegetative pole, another one is the animal pole. The animal pole is where the actual action happens. 1 to 2 cell, 2 to 4 cell, 4 to 8, 16, 32, 128 cellular stage, and gastrulation, bastulation, organogenesis process. Here you can able to see a clear visualization of zebrafish development. Its embryo developing into a larvae. So this uncoiling of tail is there. So this is called a somite formation. So each and every phase of zebrafish, its entire embryogenesis process can be marked up to a technique called teratogenic toxicity index. So teratogenic toxicity index is nothing but putting up a compound in the embryo and studying the developmental growth phase of the particular larvae or an animal with respect to its exposure time along with its developmental phase with respect to its exposure time with the pesticide or a fertilizer. So it takes 72 hours for an embryo to develop into an adult, for an embryo to develop into a fully grown larvae to extend. So during the 72nd hour or till the 84th hour, the zebrafish embryo will be coming out of its yolk sac. It will come out of its yolk sac with a velocity of 90 mm magnum pistol. So this is a resilient animal. This is a resilient animal. This is an animal which has a very vibrant development. For an example, you see as in the picture, once again I'm putting up. Okay. So you see this is an embryo which is developing. To extent people will think since it is a delicate material, it is very sensitive. Adding up the chemical will definitely kill the organism. But it is actually very opposite. You talk about zebrafish embryo, it is a resilient animal. Okay, it is an animal which creates a velocity of 90 mm magnum pistol bullet when it is coming out of its yolk sac, means it can survive to a larger concentration of your toxic chemicals to be tested. So that's why zebrafish embryo is nowadays becoming a OECD guideline model. OECD guideline model. OECD is nothing but the European Union Economic Forum for establishing protocol for testing agricultural pesticides, chemicals, and other non-toxic or toxic biohazardous materials. So zebrafish is a developmental model, a gold standard developmental model. So what and how we can utilize it? Moving ahead, using zebrafish embryo as a toxicology model for your fertilizer or a biopesticide. So what we can do? These are the OECD guideline model. Okay, as per OECD guideline model, either you can directly take up a pair of fishes, which you will be utilizing for fish embryo or fish acute toxicity. So there are different models. The common one that we are using is like 96-hour zebrafish embryo toxicity model. So you will take up a embryo at 4 HPF. HPF means hours post fertilization. So during the evening, you will be putting up a pair of zebrafish in your laboratory. You will go during the night. At the early morning, during the first ray of sunshine, definitely breeding will start to occur. Since this is a tropical animal, zebrafish is a tropical animal, it is an Indian subcontinent. There is no require for advanced, sophisticated temperature or controlled environment. Putting it in a simple tank in a closed aseptic condition is sufficient. Then you can wait for an overnight period. Since breeding is a basic instinct, it is a reproduction cycle, basic instinct. Generally, it is happened at the first ray of sunshine. So early morning by 7 o'clock or 8 o'clock or 6 o'clock, you reach your laboratory, or you have to stay night. That is up to us to decide. Okay. So you can take up the embryo. You have to take up the embryo within the first 4 hours of fertilization. Then you have to add your compound. Put it in a Petri plate, 96 well or like 24 well Petri plate, different concentration of your pesticide or a fertilizer, you put it in, you wait for 96 hours. During this 96 hour, every 24 or 48 hours, you have to take something called mortality reading. So based on the basic mortality reading, we can able to get the LC50 or like LD50, lethal dosage or lethal concentration 50 of your green nanoparticle. Like plant extract, so many people are using zebrafish, pesticides, fertilizer, microgreen algae. Even for certain probiotics we are using. But for probiotics, since it is a bacteria, it can create infection in a different way. So you have to be very careful if you are maintaining the required level of CFU, colony forming units, for probiotic based toxicity study using zebrafish embryo. And adult zebrafish can also be used, but for adult zebrafish, the ethics is stringent. You have to get a proper guidelines, like you have to get your ethical clearance. Embryo, experimentation of fishes by zebrafish, by experimentation of fishes released by CCSCA or CPCSCA in 2021 states that embryo can be taken up for toxicology study without any CCSC approval. You require a basic institutional committee approval is sufficient. For adult alone, you have to go for animal ethics committee. So there is a gap. Adult, you require animal ethics committee. For embryo, you require your institutional committee is sufficient. You can get a confirmation from your guide or HOD. You can test your green nanoparticles. You can test your newer material that you are synthesizing using greener technology in zebrafish embryo. So if you ask me a question, if we are going to go for only the basic toxicology testing, no, not only the basic toxicology testing, you can use zebrafish for multiple testing. Zebrafish embryo also, larvae also. For an example, this is neurobiological behavior testing, wherein we will take in zebrafish larvae, we will put it along with the required chemical toxicant, we will observe its movement. Okay, hypertension means bigger movements. Okay, Alzheimer we can do, Parkinson we can do, neurological studies we can do with zebrafish, but that's a different study, so that we will discuss in a different position. In a different condition. So zebrafish embryo for toxicity, apart from mortality, you can observe more than 21 different visual cues. So teratogeny toxicity index, this is what I'm talking about. Taking a chemical material, taking a nanoparticle, a pesticide, apart from saying the pesticide is harmful towards zebrafish or like my animal or to the ecosystem, I can deliberately and exclusively, I can tell what kind of pathway my nanoparticle or like my fertilizer is harmful towards the toxicity that is creating in a ecological environment. For an example, developmental toxicity. Take about zebrafish larvae. Zebrafish control larvae, you can see over here in the first picture. Okay, so you can see a fully grown larvae with a clearly developed notochord, the tail. Okay, the larvae has its own tail, which is nothing but the spinal cord uncoiling, notochord development. This is a chordata. This is like a vertebral animal like humans, right? But if you take a abnormal fish with a toxic material, the tail will not be straight. So the tail will not be straight. There can be, you can easily visualize edema is there. You can easily visualize edema, which is nothing but enlargement. Okay, and there are something called jawbone deformation is there. Here you can see, so tail is not grown, formation of eye is not there, and yeah, the tail is either curved or bent, notochord malformation. So each and every visual cues, each and every visual cue signifies a particular toxicology pathway that your chemical material or a pesticide or a toxicant is going to cause over to the environment, right? So moving ahead, zebrafish can be used for so many greener technology approaches. So this is Alzheimer model. So we don't want to talk too much about that. We will directly go on to the greener biotechnology applications. First thing, I can use my zebrafish embryo for phytochemical or like plant extract toxicity evaluation. Without directly going in for any ethical clearance, zebrafish embryo can be taken in. Till now, you can utilize it. Then bioremediating agent, utilizing, taking your microalgae, enzymes, biosurfactant, and using zebrafish as a actual testing agent. Why? Because you're going to test your toxicity. For an example, we let us create a fertilizer. We are creating a nano biofertilizer. Excellent thing. Talk of the town, nano biofertilizer. We are creating it. We cannot test its bioactivity. We can use something called seed germination test, which will give you the sprouting value. Apart from that, if you are having a regulatory situation where you have to test it in a live condition, you cannot go for a cell line test. Cell line test has nothing to do with the nano biofertilizer, right? You cannot go for rodent test. To extent, at least rodent, rat or mice is fine. But again, rat and mice is very costly. But if you take a zebrafish adult or embryo, it is the actual apt model to test your enzymes. Because at the end of the day, this enzyme is going to reach the market, and from the market is going to reach the ecosystem, especially pesticides based enzymes, especially fertilizer based enzyme. And finally, talk about you are developing a bioproduct like a food particle. You are developing a food material. You are developing a probiotic food. You are developing like for an example, there are so many food development in the market nowadays, right? So like taking in a enriched food product using a phytochemical enriched or nanoparticle enriched food product, probiotics enriched food product. And again, zebrafish can be used for a efficacy testing like dosage development, efficacy testing. Till this dosage, zebrafish is not having toxicity. The same dosage can be utilized in developing my food product. So those things, nutraceutical development, food product development, pesticide development, phytochemical bioremediating agent development, zebrafish adult and embryo can be the excellent gold standard model to test toxicity. So I'm having a little time. So let's go for other models also. Apart from zebrafish, there are few other alternate models that can also be used for green product testing. For an example, take the Venami. Venami is nothing but the king prawn. You know, the prawns that we are having in our day-to-day life, like the prawn, if you take and go to a market to take in as a food, so people use it to sell Venami, white-legged prawn. I mean, white-legged shrimp. Okay. So this Lipopotamus vinami is nothing but a white-legged shrimp, which is one among the common product which is available as a food, especially export food product in the east coastal region of India. You talk about Vishakhapatnam, you talk about Chennai, you talk about Toothukudi, you talk about some place in Andhra or like Odisha. So many people are growing shrimps, developing shrimp for food product export, but the same shrimp can be utilized as a model, especially for greener products. So, what kind of greener products you talk about? Like anything pertaining with aquaculture. You are developing a product for aquaculture. You're developing a food for aquaculture. Okay. So greener product development involves like conversion of waste to value. So many people are taking in waste, they are converting fertilizers and pesticides, and some people are converting waste to livestock feed, right? So you are converting waste for poultry feed, you are converting waste for like a cow or something larger and higher animals, but you cannot always get the luxury to test it in this animal. Let us assume you are developing a food product for like your poultry. You cannot take in 10 chicken, or you cannot go with the cam assay, chick embryo assay. You need ethical clearance for that also. But you can take in your Lipopotamus venami. Litopinamus venami, which is nothing but your light white-legged shrimp. You can take in the shrimp, you put it in different tanks, you study its toxicity. So using shrimp as a toxicity model for aquaculture-based products, plant-based feed additives, environmental pollutant monitoring. Because at the end of the day, the environmental pollutants, we are talking about sustainable greener technology, but when you talk about environmental pollutants, after putting it in a common effluent treatment plant, it has to reach up to the environment ecosystem, like put it in a river or a pond or a ocean. But to study the actual ecological impact of this treated or non-treated or semi-treated, for example, we had a wonderful previous speaker who was talking about bioremediation. We are developing a bioreactor for so many purpose. Excellent presentation, right? So you talk about bioremediating, you put in your bioremediating agent like an algae or a nanoparticle or something like that. You are treating it. After treating, what we will do, we will put it in a UV spectrophotometer to study like the output, like whether the dye is break down into the required particle. Hmm? But now what we can do, we got like some other studies in our laboratory that I have guided as a PhD guide. What we will do, we will develop a bioreactor with the client specification and requirement. And every 1 hour or 3 hour or 5 hour after the treatment, we will take out the extract. We will take out the treated effluent from the bioreactor. We will put it in zebrafish embryo or we will put it in the shrimp and we will study the toxicity. So increased time in the bioreactor, there should be a decreased time towards the toxicity output or like the mortality rate towards your animal model that you are testing. So utilization of shrimp or zebrafish embryo for toxicants. So this is one way where you can take it in. One more model that I want to conclude with. So this is Artemia salinea, which is a common shrimp. This is called as brine shrimp. Have you heard about brine shrimp? Brine shrimp is nothing but small white color dot. The initial that white-legged shrimp is very big prawns, right? It's like a king prawn. Whereas this shrimp, this is a very small in structure. The name, colloquial name is called as sea monkeys. I would like to share a small video where you can able to observe it. Just a second. So you talk about this one, you know, these are nothing but very small animal, which is called as sea monkey. So this is the developmental phase of this animal. Okay, you see. Hmm? So it has just two wings, which will be flapping in opposite direction, and the movement will look like a monkey moving from one tree to another. The basic advantage is that brine shrimp can be cultivated anywhere. You talk about larger shrimp, you require a bigger laboratory to cultivate. But you talk about brine shrimp tablet are even available in Amazon. You can take up a brine shrimp tablet, you can put it in a small pot, you can put like a warm lamp, sodium lamp is there, right? Similarly, like a warm lamp, you can take it up. Okay, within the first 12 to 24 hours, it will hatch. The brine shrimp egg, which is available as a capsule in Amazon, each capsule costs around like 5 to 10 rupees. It will hatch like within a few minutes. So what it has to do with greener chemistry? It's very simple. Utilizing this as a model for studying your bio-efficacy or like toxicity of your pesticide and fertilizer. So the basic advantage is that this is a model which can be easily developed. This is a model which can be easily developed within like overnight period. Okay, and the cost is also very effective. But the basic advantage, you can study nanoparticle toxicity, you can study pesticide, the same thing that you are going to do with zebrafish or like the king prawn. The only requirement is that king prawn or like zebrafish requires a bigger laboratory or like a little more technical validation. Whereas you work with brine shrimp, anyone can work with brine shrimp. You can take up a tablet, you can put it in water. Within 12 hours, you will get small animals. You have to count 10 animals per well. You have to put 10 animals per well. Then you put it in your product. You test its bioactivity. Okay, so why we require this animal model? We can develop a dosage dependent non-toxic material in comparison with the pre-existing toxic material. Talking about greener chemistry solution, talking about dye treatment, talking about nanoparticle or other material development from a green perspective, we need to know the dosage, the actual dosage which is non-toxic or actual dosage which is less toxic. What is the threshold limit? Every time for a threshold limit, you cannot go in and put it in a larger animal model. You take in brine shrimp. With brine shrimp with 96 well plate, you can study up to 96 different concentrations or dosage within 12 hours. So whether this dosage is toxic or not, how you can do, you can put in the dosage, you can put in your animal. More than 50% of mortality means that particular dosage you cannot take. More than 80% of mortality, never consider. Okay. If it is having less than 30 to 20% of mortality, you can grow again, work it on, you can reproduce, and you this time you can test it on a higher model. So what we are suggesting is that rather using traditional higher animal model, initially you can develop your greener biotechnology products and test it in alternate or lower animal model. Then you go for a optimized material to a higher animal model. One last model I am going to give a glimpse of is Drosophila. You know, the common fruit fly that you see it in your April, May in Indian subcontinent, right? There are different types of fruit flies. Bigger fly is there, small fly is there. Small fruit fly. Okay, this is not the house fly that I am talking about. A small fruit fly that you can see in vegetable shops, fruit shops, like in a normal fruit shop, right? Okay, so what is the advantage? The basic advantage of utilizing Drosophila as a testing animal is that it is very, very, very sensitive to pheromones. So gaseous compounds, volatile gaseous compounds. So Drosophila is a common volatile gaseous compound testing animal, right? So I don't want to take a larger time. Just one more small video I will show you now. Okay. So this is the animal which will mate for life. So this is the animal which will mate for life. That means zebrafish, I mean, Drosophila is an animal which is very, very instinctive towards selecting a partner. So this is the courtship behavior of a Drosophila. So where you can see the female would be there, the male would be there. The male will like move around the female in terms of development. I mean, courtship behavior. And how we can use this animal is that, okay, so many reproductive biology related materials or like the toxicants are generally gaseous in state. Okay, you cannot take a gaseous material, study it with a rodent. Even in zebrafish also it is harder to study it with a gaseous material. But you take a gaseous material, you develop a small closed chamber for the Drosophila, you inject in different conditions. Okay, increased pheromone means increased courtship behavior. Increased pheromones means better toxicity or better efficacy. So toxic gaseous material can be studied, especially which is related towards reproductive biology, which is related towards pharmacology aspects of reproduction. Those materials are still now tested using Drosophila. You talk about Drosophila, you have to talk about the courtship behavior. So the courtship behavior means it is very, very specific towards the required male and female pair. If the male and female pair is released onto an environment with the required gaseous condition, it will mate. This gaseous condition is nothing but the volatile compounds that you are studying for, which you can take in as a alternate model to develop. Like there are drugs which will increase the reproduction rate, fertilization rate, like for erectile dysfunction, there are no bigger model to study. You cannot study it with mammal. You can use Drosophila. But especially for greener applicants, what we can do is that we can take in our gaseous compounds, we can study its nature. Especially you talk about pesticides. Most of the pesticides that we are taking in are like generally gaseous in state. Okay, you're developing a nanoparticle, you're converting it, you're putting it in an atomizer, you're developed as a gaseous product. Now you have to test. This gaseous product cannot be even tested in zebrafish model. At that time, you can use models such as fruit flies. So these are various models that you will be utilizing to develop your greener technology applicants or products. And mostly it will go around toxicology. Any model that you will take, any product that you will take, initially you have to test its toxicology, like the LD50 value, IC50 value. Then the established LD50 value, IC50 value, from that you can create a threshold. From the threshold, you can create the required bioefficacy dosage. Like 50 mg is my LD50, means 5 mg is my LD10. LD10 is the highest dosage level. From LD10, I can create pharmacology viable dosage, or like pharmacologically useful dosage, which can be taken up for bioactivity prediction of the same material at a lower level, which is non-toxic. So I hope this would have given you an idea. Maybe this is little towards out of the theme in certain aspects, but utilizing alternate animal model in terms of greener product development will pave a better society, will pave a better technology, will pave a better ideas for sustainable research practice. So I guess this is what I have prepared. And with this, I can go and question if there is exist any. Yes, ma'am.
Thank you, sir. Always animal model are very interesting to everyone. So it was nice presentation. So a few questions from the participants. That one question you have already responded for how reliable these models are. So next is nothing but like how do we choose the appropriate model for specific diseases, sir? How do we pick which type of organism can be used or tested for specific type of diseases? Because a number of diseases are coming in place now, blooming it up. So how do we fix it? How do you or how the researcher or scientist will fix which type of microorganism or which type of organisms can be picked for this type of modeling testing?
Yeah. So yeah, each disease model require a particular data point to be observed. For an example, initially we were talking about zebrafish. 70% of the protein coding gene present in human beings are also present in zebrafish. Even though zebrafish is an animal which is present in a different ecosystem, it's an aquatic ecosystem, it's living inside a tank. We are like an air breather, we are in an ecosystem which is entirely different. But more than 70% of the protein present in zebrafish. So that means we can use to extend, but again, in terms of selecting the model, not every disease model in mammalia cannot be replicated in zebrafish as a host organism. For an example, any reproductive model, it is harder to develop because mammalian reproduction pathway, mammalian reproduction output is entirely different and requirement is entirely different when you compare it with the fishes like zebrafish. But when you talk about neurology, zebrafish is the gold standard model for Alzheimer, Parkinson, as well as you talk about hypertension. One particular example is that zebrafish has an excellent learning and memory disability. For an example, you take a zebrafish, just put it in retro perspective in a minute. So you take up this is anxiety model, this is Alzheimer model. Okay, you take up a zebrafish, you put the fish in the middle, T maze. Every time you have to train the fish. Left means reward, right means danger. Reward means very simple. You can given the food. Right means danger means every time you have to pinch the fish when it comes to the right side, or you can create discomfort in terms of swimming. Right. So eventually, ultimately, zebrafish is actually an excellent animal. You talk about other fishes, it doesn't know the boundaries. You put a tank, you put a goldfish, it will hit on the boundaries of the glass tank, right? Whereas you take a zebrafish, you put it in the water, it will never hit on the glass tank of the container. It will never hit on the boundaries of the glass tank. That means zebrafish as an animal is a sentient animal. It knows the glass boundary. Not only glass boundary, it has an excellent optokinetic response. It knows visual cues like the right color, bright color, dark color. So red color means danger. Blue color means or green color means reward. You train the fish. Once you have trained the fish, there is a chemical compound called scopolamine or ocodoic acid. You give that chemical compound either orally, we can give zebrafish oral gavage also. So either orally or like you can put it in the tank water also. Different dosages is there. You put it there, then definitely the same fish which is trained will develop a Alzheimer-like condition, memory disimpartment, something like that. Then the same fish, if you put it in the again same T maze, what will happen? It will not know where to go. It will not know where to go, like right or left. So once an animal, a trained animal, which is an intelligent animal, which knows the food, which goes to the food, after putting it in a chemical, this time it will not go to the food. So this is Alzheimer-like condition. Then you put your drug. You put your drug in 3 concentration, low, medium, high dosage. So what will happen? High, medium dosage, if the condition is reverted back, like now the animal is knowing where to go in terms of memory, means you can take in. So each disease requires a particular data point. Any particular neurological disease, neurobiological behavior is the data point. Zebrafish behavior can be maintained. Zebrafish behavior can be tamed. Like you can train the fish, right? Then again, zebrafish behavior can be monitored using a, for an example, a recorded video, put it in a software. So neurological disease can be studied using zebrafish. Alzheimer, Parkinson, anxiety. Anxiety is an excellent condition. You talk about anxiety, you talk about depression. Depression is an excellent condition. You talk about epilepsy. Depression, let's talk about depression. Depressed zebrafish, what it will do, it will go to the end of the black tank. End of the black side of the tank, it will move, it will not move, it will move towards the end, it will not show any movement. A normal zebrafish is a happy animal. It will have a pole-to-pole swimming, left side to right side. That is the common normal behavior. Every time you put a zebrafish in a tank, it will move to the left side to the right side. But a depressed animal will not move. Then you put your antidepressant into the study. Antidepressant means elevation of mood. Better moods means better swimming pattern. So within the swimming pattern, neurobiological studies can be observed. So zebrafish can be used for diabetes. Diabetes is that you take blood at a different condition, you observe the glucose level of the blood using a normal blood strip, glucose strip is there, right? You can observe. So, but neurobiology, the basic obesity and like diabetes can be done. Cancer studies, it hard to do with zebrafish because zebrafish, you cannot maintain a particular cancer. This small animal with 2 inch of growth. Within a 2 inch of growth, you cannot go for a organoid based cancer like liver cancer, hepatotoxicity. You cannot develop because generalized cancer model can be done. So organ specific cancer model cannot be done. It can be done, but it is a harder condition. So zebrafish model, neurobiological condition can be done. Diabetes can be done. Obesity can be done. Similarly, you talk about brine shrimp. Brine shrimp is a very small animal. You cannot do neurological studies. You cannot do bigger studies. So brine shrimp is for acute toxicity. Brine shrimp is for acute toxicity. Shrimp is for chronic or subchronic toxicity. Zebrafish is for neurobiological study. And finally, Drosophila is for behavioral related study towards gaseous environment. For an example, Drosophila, I have even put it in the slide. Drosophila is mostly very important for sex-linked the behavioral study. There is a particular sex gene which increases the efficacy of the animal movement with one another. Right. So each animal, based on the data point, you have to observe. So cancer, harder to study. Neurological study, definitely easy to study. Toxicology, this is the gold standard. You cannot go for rat or mice for toxicology, especially for pesticides or fertilizer. So you have to take an aquatic model. You have to take an amphibious model wherein you can study your toxicity using this the current model that we are discussing. Maybe this gives an idea, ma'am. Yes.
Thank you so much, sir. You have answered for all other questions which is asked, like how effective it is in cancer and toxicity test, everything. So anyway, thank you for your valuable input for this webinar session. And on behalf of Karunya Institute and IEI Coimbatore Local Center, I thank you, once again for your accepting invite for this webinar. Thank you so much.
Yeah, ma'am. Thank you. Thank you to Karunya Institute and thank you to Institute of Engineers for this wonderful opportunity. And I'm sorry I've taken a little longer time. Generally, I will become very enthusiastic when I talk about our animals in research. Once again, thank you all for your wonderful participation. Thank you, ma'am. Thank you.
Thank you so much, sir. Now I am happy to invite next speaker of this webinar, Dr. Manikandan Maharaja, is a researcher researcher and innovator in the field of green biotechnology and sustainable materials. He completed his PhD in 2024 from the Department of Green Energy Technology at Puducherry University. He is currently the co-founder and director at Xenobiotic Research and Technological Development Private Limited, where he leads cutting-edge research in environmental and sustainable technologies. His work focus on integrating advanced 3D printing techniques such as FDM and directing writing with green biotechnology for application in environmental remediation, energy system, and biomaterials. He has developed novel 3D printing photocatalyst for wastewater treatment, bioinks, and biocombustible polymer system. Dr. Mani has also designed and developed the customized 3D printing system, including a DIW-based bioprinting and a filament making machine for which he holds a design patent. His interdisciplinary research spans nanomaterials, catalyst coating, biosolar system, and environmental sensing technologies. His contributions have been recognized through the multiple projects and national level innovation platform, including collaboration with IIT Patna, NIT Calicut, and the Texas Instrument Innovation Challenges. Today, we will be speaking on 3D printing enabled green biotechnology for sustainable environmental remediation. With this intro, I welcome you once again, sir. The session is over to you.
Thank you, ma'am. Thank you for your nice introduction. I will share my PPT.
Yes, sir.
My screen is visible now?
Yes, sir.
Yeah. So, good afternoon, everyone. Thank you for a warm introduction about me. So today I'm going to taking about the topic is in 3D printing enabled in green biotechnology for environmental sustainability. So basically, we are focusing on environmental challenges and limitations and green biotechnology concept. So in environmental challenges like water pollution, air pollution, and soil contamination and industrial waste, in this problem facing, we are expecting the limitations of conventional method for high cost and low efficiency, secondary pollutions, poor reusability of the materials. So for the basic, we are taking the solution in green biotechnology for microbial degradation, enzyme catalysis, and eco-friendly system. So in that, we are taking into the application for 3D printing for that green biotechnology. What is the 3D printing? So about 3D printing, 3D printing is a three-dimensional object in its physical configuration from its digital form, from layer by layer adding form. So you can see that a CAD file we can design in a model, and then we convert it into the G-code file in the 3D printing slicing software. Then once we loaded that file into the 3D printer and start, then we can get the final, our physical object. So for in 3D printing, recent trends in many applications we are using. For example, 3D printing ultralight aircraft parts we can use. Animal parts can help reduce the black market rate, and frictionless gear could revolutionize space application. Wood waste born again a 3D printed furniture, and Swiss MEMS chip enabled 3D printings and prosthetic eyes of the futures 3D printed, turning waste into walls with additive manufacturing. So transform your 3D printer into a versatile paste extrusion drive. Yes. So like many various application recently we are focusing on. So overview of 3D printing technology, you can see in that images for this is a one patient 3D model, scanned model. We can convert it as a STL file, and then we can print it in the layer by layer. So this is the before printing, we can check in the software how the printing formation will come. So when this is the real model, but while we printing with, we are generating the support with that model we are printing. This is the basic of that understanding of 3D printing. So when we talking about the 3D printing, so there are multiple types of slicing software is available in the market. So for example, it is available in paid and open source. For basically in open source, Ultimaker Cura these all are the promisable software. Many hobbyists and researchers are using these softwares. So further, that classification of 3D print technology. Based on that, 3D printing we can classify it into that 7 types of applications. So on that, we can, based on raw material, we differentiate 3 types: solid based and liquid based and powder based. In solid based, we will here discussing about fused deposition modeling, and liquid based is a resin 3D printer, and this all are powder based for select SLS and direct metal laser sintering, selective laser metal sintering, and electron beam melting. And based on layer formation, all the 3D printers are adding a layer by layer, but these applications based on that using materials: metal powder, plastic material, ceramic material, and thermoplastic material. So this is the 7 types of 3D printing technologies we can use on based on our applications. So when we're talking about that 3D printing technologies, when 3D printing, we are using the material. So here, list of material choices is available for 3D printing. So in 3D printing materials, for DIW material, it's a direct ink writing materials. So which is, we can make a ink, bio ink for various to print a ceramic material, tissue engineering, bioscaffold, and then a printable electrodes. And then metal 3D printings, we can print it for aerospace applications and defense ships, motors, all that. Very difficulties, motor blade shafts, these all the components we can printed using the metal 3D printing. And then other alternative method is that FDM material. So this material is this available in various types of thermoplastic material like polylactic acid for PLA, PETG, and then carbon fiber reinforced material. These all are the biocompatible material. So when we talking talk about the direct ink write technology, DIW. So which means the DIW printers enable the precise deposition of functional inks to create complex 3-dimensional structure. It's offering significant potential for the fabrication of advanced devices and components. While the print bed area, motion controller, and slicing software is common as that of FDM printer, the printer header nozzle design are exclusive and specialized to ink write printers. So this is that dimension view of that Bio 3D printers. And this is the design and fabrication of the DAW printer. So you can see video. Okay, video is not playing. It's technical problem is there. And further that FDM 3D printer applications in fuel cells. It's we are talking about that case study of that applications. Fuel cells for types of fuel cells, low temperature and high temperature. Based on that, here proton exchange membrane fuel cell and direct methanol and ethanol fuel cell, alkaline and phosphoric acid and bio-catalyzed microbial fuel cell, and high temperature molten carbonate and solid oxide fuel cells. So in that fuel cell, it is a low temperature, highly energy content, low toxicity. Ethanol is the ideal fuel to give fuel cells. So basically in fuel cells, in standard, we will use that bipolar plate channel. So this is a conventional method. They are making these components is very traditional method for molding and then to making that constant fuel cell dimension channels. So when we making this kind of models, so but that cell cell efficiency will be drastically it's not that increase the performance for standard convention method. When we here replacing that 3D printing, when we are using, so instead of that using the conventional method for that high molding technology, we can custom design our freedom of design the 3D printing. So will be support with that we can printing and then fabricating our this complex geometry model design using the 3D printing technology. So in this fuel cell performance by optimizing the flow path, channel geometry, and pressure flow parameters are presented. While basically we are talking about the fuel cell designs, they are connected with 9 types of components for anode and cathode sides. So to make a sandwich, and to anode side we are passing our ethanol fuel as a biofuel, and cathode side is a air passing. When we purging that ethanol, then reaction will be started. It will be converted that hydrogen and proton and to produce that electricity. So this is the schematic proton exchange membrane fuel cell. This is the dimension we are taking consider in this fabricated cell. So basically, when we are going to before that making any fuel cells, for cost wise we have to reduce. Before we are using that simulation software. Before that, using that ANSI simulation software, we will design that multiple designs with that standard parameter literature review studies. We will consider into that factor, and then we will take in this is the channel depth parameters, and then we will measuring that boundary condition, physical boundary condition with that regarding to the literature. So finally, we will get that result. Based on that simulation result, we are considering which is the best optimization design. So we will varying with that here different design for interdigitated and then single ladder and double ladder, and then various cross section dimensions with respect. We are finding the best current flow channel, and then varying that depth of the channel, and then effect of pressure we are changing, and rest of the physical parameter condition we set as a same to getting this result. So finally we are getting that result in that single and double ladder, which is the best current density is giving that maximum cell. So according to the design, we can using the simulations method to find that best design model. Then we can go for that 3D printing, fabrication testing of the fuel cell flow path. So 3D printer flow path plate, weight and reduction strategies we are considered. Before we are talking about that conventional method and then 3D printing method. Here 3D printing method is the advantages for free freedom of the design and cost reducing technology. Instead of conventional method, if you are going, that material weight is 100 gram, it is there. If you are using the 3D printing technology, here that is advantage is we can use that lattice structure printing support. So with the technology, we can use minimal material, but that same mechanical strength we can attain. So without loss any operational losses, so that we can attain. So for that we consider this is all the parameters we are considered in the software before we are printing. So you can see when we talking about the 3D printing, what is the 3D printing in between that how the material is filling. So we can use that infill area we can tell. So this is like a difference rectilinear, fast honeycomb, full honeycomb, grid, triangular, and then wiggle shape. These are available in this software patterns, automated. So with that, so once we design the model, so you can see in that G code view how before printing our model will be representing in the software. Then finally our 3D printed cell, it's showing in that figure DEF. And then this is the standard graphite cell by conventional method fabricated for figure G and H. You can see the weight of that standard graphite cell is about 80 gram, and then our 3D printed same dimensions with that optimized channel design, but we are reduced to 1/3 of the weight, around 23 gram. So these are the advantages of the 3D printing. And then here, when we consider about the full cell assembly, so for standard fuel cell weight is 979 gram. So when we used for that 3D printed bipolar plate, it's coming to 866 gram. So weight reduction is 11% around it is reduced. We consider about the only flow path plate, it's benefited about that 70% of the weight reduced in that bipolar plate instead of that conventional method manufacturing 3D printing advantages. So this assembly of fuel cell, we are connected and leak test and then design optimized 3D printer flow path model weight is compared with the standard cell. So it giving about a 70% weight reduction is achieved. Overall, 11% weight reduction is obtained. So this is image, you can see that a 3D printed fuel cell assembled and then checked with that fuel leakage performance. So this is the publications work with that 3D printer. And when we come into that another, so this is what environmental remediation for catalyst coated FDM 3D printed matrix for environmental remediation. So for basically conventional catalyst substrate generally made in standard rectangular shape as limitation. Following this 3D printing technology, it's a design freedom. One can print it in complex surface enhanced lattice structures by design optimization for better performance. So for here, what we are expecting the novelty, it's a custom designed FDM 3D printed honeycomb substrates we can modify to absorb the nanomaterial catalyst. When incorporated into the photocatalytic reactor, can successfully mitigate environmental pollution on a large scale. So custom design FDM 3D printed photocatalytic honeycomb structure is coated with a pre-synthesized visible light active nitrogen doped TiO2 catalyst. Optimization of different types of solvent was conducted to identify compatible solvent that can retain the efficient functionality of NTiO2 immobilized onto the PLA substrate. So here we are taking this material and equipment. This is that methodology of that process of that making N-doped TiO2. So this is how in figure representation you can see that left side figure ABC. So 20% and 30%, 40, 50, 60. It's a complex lattice structure. So this all are by tricky. We can easily made it in that 3D printing software. With that infill structure, we can use this complex honeycomb structure. So, but before when we making a design 3D model, one solid round structure, we will design in 3D modeling software, CAD model in figure A. But when we printing into that figure B, using the 3D printing software, we can using this infill pattern orientation with that honeycomb structure substrate we can print. And then finally, figure C, you can see the 3D printed honeycomb substrate. So this all the 3D printing parameters when we are considering before printing, we have to consider in that software. So each printing technology, when we are before printing, we have to consider that the temperature of that nozzle and then bed temperature. This is based on that when we are using that our raw material. So here we are used PLA. If suppose we are using PETG and ABS material, it will be coming in a different temperature settings. So for this technology, it will be that printed substrate we taken and cleaned with that ethanol. That matter is cleaned with ethanol and dried at 60 degrees Celsius for 5 to 10 minutes in an hotter oven. That 3D printed honeycomb matrix was immersed in coating solution for 30 minutes. The coated matrix was allowed to dry at room temperature for 30 minutes and then kept in hotter oven at 60 degrees Celsius for 12 hours. And the coated substrate improved its appearance. You can see in that images. So this photocatalytic experiment was performed to assess the functionality of the prepared catalyst. So for that 0.5 milligram of methylene blue taken in 100 ml of water, the 3D printed matrix catalyst is immersed. The dye solution is stirred magnetically for 1 hour at 160 RPM range. The dye solution with coated substrate kept in the dark for 1 hour for magnetic stirring to arrive at adsorption-desorption equilibrium. A 2 ml of dye sample was drawn every 30 minutes interval. A sample was analyzed 400 to 700 nanometer absorption using a UV visible spectrometer. So in that sunlight conditions, so far according to that result we are confirming base TiO2 material and then NTiO2 material confirming with that synthesized NTiO2 presence in XRD and then XPS material and same images. So every 30 minutes when we sample we taken, before that we prepared the TiO2 and NTiO2 coated onto that substrate. We are taking that suitable additive solvent. So here we are taking that 4 types of solvent, toluene, ethanol, and then tetrahydrofuran. So with that different types of solvent, which is giving the best result, we are optimized and then attaining. So in that experiment, extent of degradation in methylene blue dye in 3D printed honeycomb catalyst. So what here we are talking about, in generally conventional method, basically in research, TaO2 catalytic wastewater dye degradation, when we taking about that TaO2, our synthesized nanoparticle powder, so we will put it into the beaker using the vigorous magnetic stirring. Degradation is done now. Then the retrieving of the catalyst is lost almost. We cannot retrieve for 100%. So for that to replace that difficulty and wastage of the catalyst material, so we have deposited that catalyst material onto the 3D printed substrate. We are using that number of cycle repeatability is increased here that. So that is the advantage for 3D printing substrate. 3D printing is support for that complex structure. Using that substrate, we are coating that material onto that and then using number of cycles to get that a cycle degradation from first cycle to fifth cycle, about 50% degradation we are attaining multiple times. So you can see in the number of cycles repeatability we are done. So from 90 to around 70% in third cycle we are getting. With respect to that material is coated material removed from that substrate. Also losses is there. So these all are we observed on that performance of the material. So the design optimized FDM 3D printed PLA honeycomb structure can act as a good substrate holding photocatalyst for water remediation application. The toluene assisted N-doped TaO2 catalyst successfully incorporated into 3D printed PLA matrix for application as an alternative to powder catalyst material. The cyclic test of dye degradation by PLA matrix catalyst shows satisfactory stability, retaining up to 50% of loaded catalyst on the material. On that next, we are going to talking about design and fabrication of 3D DAW printed TaO2 and NTaO2 catalyst matrix for environmental remediation. Before we are talking about taking a FDM 3D printed substrate, we are taken the TaO2 and NTaO2 catalyst coating onto the substrate and studied that dye degradation method. Here, directly we are taking TaO2 and NTaO2 100% as a green ink, green synthesized ink material, directly printing and then cure, and then we are using 100% catalyst material. So this is a DIW method, and custom design and fabrication of continuous photocatalytic reactor we are done. This application of 3D printed TaO2 catalyst in rhodamine B and methylene blue degradation we considered. So XB 3D direct ink writer, this is a customized 3D printer developed by us. And challenges in industrial application of photocatalyst. Initially, I talk about that generally. For what is the challenges? Basically, here again, it's a photocatalyst applications wise 3D printing tools we are using, textile dye wastewater, TaO2 and NTaO2. Basically, in research problem, band gap, we are improved by that material synthesized method, nanomaterial, and then recyclability. This is the research problem. So in that recyclability, how we can attain? So for that, we are using the 3D printed directly. So number of times recyclability we are attained. So state of art, this nanomaterial yield and economy, slim membranes using coating method technology. So our solution, novel 3D printed designs, we are incorporate. So in that photocatalytic reactors are essential in promoting sustainable and efficient catalytic process. The system can accomplish effective dye degradation by continuous process. So this is a reusable 3D printed catalyst substrate. So these are that materials and method we are used in there. So this is that schematic of that how the process from initial to end we are making. So taking the catalyst, grinded very finely and make that ink slurry paste and loaded into that syringe, then fixing onto the 3D printed cartridge, then we will print it according to our given dimensions for TaO2 and NTaO2. So this is the custom design of batch photocatalytic reactors with that rhodamine B dye. So this is how that continuous photocatalytic reactors we designed modeling. So this is that reusable 3D printed catalyst substrate. We can make a custom photobioreactor and making a stack arrangement. Then we pour our methylene blue dye with that light condition. With that, we every 30 minutes we are taking the sample, then we will get our result and discussion. So this is the topology optimized design and fabrication of 3D printed TiO2 catalyst. So in that figure A, B, C, D is that we can design the catalyst substrate before printing. We can see in that slicing software. So this is how it will become. Before printing, we can check how that pattern formation will come. In that figure E, F, G, H is that after printed, our post-cued catalyst material. So this is all that dimensions after printing that printed catalyst weight material you can see in that table. So this is that side view of that 6 layer of the 3D printed layer by layer TiO2 material. So this is printed by 0.8 micron nozzle diameter. So total height of the material is 5.10 mm. Each layer will be coming around 0.8 micron. So when we're talking about that ink, all those things, it's multiple times of trial and error method only we are optimizing and finding. Because when we am talking about before that FDM printing is a thermoplastic material. It is standard optimized stock material. So we can directly load it into that. In 3D printer, we can directly print it, our basic substrate. But when we are coming into the DAW printing, so this ink material is a completely R&D basis. So each time we are taking the new our nano materials and bio ink optimization additive adder, we have to optimizing it and lots of trial and error, which is when it that better post curing method is getting. So here we are taking that one biopolymer. So carbopolymer 940, with that we printer, that carbopolymer 940 is having a shrinkage property that ink is not suitable for this material. So when we facing the problem, then we are finding that which ink polymer is alternate suitable. Then alternatively, we did a literature, then we find about the pluronic F127. With that, we added TiO2 and NTiO2. Then we will printed the specification dimension. After that, printed green cat. This is the green catalyst ink in the catalyst image. Left column you can see. After printed one, we can sintering condition. You can see that post treatment after in the right column of the images. So 3D printing demonstration how we can printing. You can see in that video. So before I can tell you that material we can grind it and load into the syringe. Once we loaded that machines, what we given, so fully in that software, it will be once we given our parameter before printing, we have to check all the parameter. Once we given, it's control taken by the machine until we have to wait and observe the printing finishing and then coming. So once layer is completed, then it is going the second layer. So this is that way that 3D printing is working. So in that application of 3D printed TiO2 photocatalyst, so here you can see in rectilinear shape and then honeycomb shape we are taken 2 patterns. So with that honeycomb shape and then rectilinear shape, so in that shape itself, we can increase that surface area of that material. So that is the another advantage of the 3D printing. So compared to honeycomb shape, that rectilinear shape is increased the surface area of that material and degradation efficiency is increased for 45% compared to the 37. This is that various article literature paper related. With that, in the present study, 3D printed TiO2 37 to 45%, 30 to 45% we are attained. So here they are using TiO2 nanotube material. So here basically when we talk about the 3D printing, so 3D printing when we are coming into that, we are producing material, we use the bulky material, large type of material we needed. So before, this technology is very new. When we are preparing that nanotube material, when we are producing 100 gram is so difficult. So what we are a solution we find it, we taking the basic material, stock material of that TaO2. First of all, the problem we have to finding out, we are optimizing that ink material. So for that, with the basic material, we optimize the ink first, and then with that basic ink material, how that material performance is given. That is what we focused on that. With that basic material itself, ink optimization is done. We are attaining 37 to 45% and then 30 to 45% in the Rhodaben B condition. We attain. So this is with the 3 cycles, we are still getting this degradation rate. In Rhodamen B, same procedure we protocol followed in continuous reactor in sunlight condition. Every 30 minutes, we are collecting the sample and taking that UV absorption. And then with that following degradation result and triangular honeycomb, we are getting that cycle efficiency. So this all the parameters, you can see that efficiency will be, variations will be there, 42, 32% and 25. When we come into that honeycomb, it will be showing that 78 and then 35, 30. It's because of in that sunlight when we are doing, that time that cloud formation will come, that sunlight intensity is reduced. So based on that, that efficiency problem is happening. So that we will taken into that after lab condition in direct light condition also we did an experiment. Here we are talking the continuous reactor for rate constant per minute, how much it is degraded. We can get it there. So then that light condition, we are not getting proper weather condition. So we moved that into that lab continuous in light condition. We did in methylene blue, same catalyst with the same reactor, but in TaO2 degradation rate is 24 and NTaO2 is 60. Why that difference? It is there, no? So TaO2 is a normally anatase fed. So it's having a less active surface area. So to increase the performance, we are using for NTaO2. So it is having a high surface area to show the better degradation performance. And then each cycle, we are continuously did with the 3D printed catalyst, that weight loss is also very negligible. So that here repeatability of that catalyst material is attained successfully. So this all the characterizations we are done in the epitated catalyst material, we confirmed by FTIR and then Romanship. So there have been notable advancement in material innovation, specifically in their creation of photocatalyst based on 3D printed TaO2 and TaO2. Known for this photocatalytic characteristic, has been a central focus in pursuit of efficient and sustainable technology. By using NTaO2, the photocatalytic performance is significantly enhanced, allowing for a broader range of light to be utilized and enhancing the separation of electron hole pairs. The incorporation of DAW 3D printing technology in manufacturing, this photocatalyst provides a novel aspect, enabling accurate manipulations of the material structure and morphology. This discovery shows potential for radically transforming the design of photocatalysts, tackling issues such as optimizing surface area. So then another applications of engineering, MXint incorporated with the NTaO2 composite photocatalyst for high throughput reduction of CO2. So you can compare this all the comparison for TaO2 and NTaO2 for CO2 reduction, structure and catalytic advantage for bulk TaO2 and NTaO2. We are reducing the band gap energy from TaO2 3.2 is basically, and NTaO2 we are reducing to increase the performance. So we taken 2 methods synthesized, urea and the solvothermal method. So this is that way of standard laboratory method for preparing, and we confirmed that material synthesized the MXin and MXin incorporated material with by SM. And this is the schematic of the DAW printing catalyst for CO2 RR. So common 3D printing techniques is DAW, FDM and stereolithography. Here we are used DAW. TAW advantage is when we synthesize our green ink nanosynthesis particle 100% directly, we can use it with less chemicals, direct green ink material, we can print and attain that result. So these are XRD analysis confirmed our material presence, and then TG analysis for the printed catalyst substrate. So that ink preparation for DAW, MX and NTA catalyst with the pluronic, and then solution prepared, and then we added that NTiO2 MX powder. With adding that slurry, we loaded and then printed catalyst substrate. This is that add dried substrate, then sintered substrate weight. We are weighed and compared it. And this is the weight loss of the material with varying temperature, sintering temperature 200 to 50. Before we are TiO2, when we sintered 700 degrees Celsius, but when we coming into the here, the same TiO2 and TiO2, we are reduced the temperature because here we are adding that MXing. So MXing property, if we are going above 400 degrees Celsius, MXing active will not be present. For that limitations, we are reduced that sintering temperature below 400 with that MXing composition. So this is how the photocatalytic CO2 reactor setup we are done. In that direct sunlight experiment, every 30 minutes, we are collecting the sample and UV visible. So with that respect, you can see in that production rate compared to TaO2, that energy band gap is reduced in NTaO2, so performance is increased. And then furtherly, we can add that MX in with that NTaO2. It is further 4 times that production rate of that material degradation is increased. So that ethanol production rate in TaO2 is 87 now. In NT MX is 774 we are attained per gram catalyst. So when techno-economic analysis for high ethanol yield, we are achieved with MX based catalyst, especially in immobilized system, but current lab scale setups remain economically unviable. That electricity and catalyst cost are the primary expenses. Shifting to solar power and reducing MX loading can significantly improve feasibility. So profitability is achievable within 3 to 5 years through scale up, targeting the high value of C2 plus product and adopting continuous flow reactor designs. So then another application is in DAW 3D printed electrode for energy storage applications. In this research, the investigation into the strategic combination of lithium cobalt and then dioxide and cobalt oxide and graphite as active material along with supercarbon as the conductive material for electrode application is made. The main focus of the studies was to demonstrate the application of the developed DAW printer on another energy storage system. For the systematically optimized and ink formulation specifically designed for the 3D printing of electrode, show proof of concept on the production of printed electrodes in characterization and application coin cell device. So these are the electrode materials we are consider to make that electrode print optimizations. So this is that 3D printed electrodes for energy storage applications. In electrode A cathode, B is anode, and C is the supercapacitor application with the following printing size and printing pattern. This is figure A, B, C. You can see that figure A is that battery cathode, and figure B is that anode electrode, and then C is that supercapacitor electrode. So the optimized ink we printed on the pattern. Then finally, we can fabricate that cell with that coin cell and to run that full cell charge studies to get that full cell discharge cycle up to the 5 cycle, we can getting the constant charging and discharging. Then that fabricated coin cell with glowing for certain hours with that in lab condition with that LED. So these are all the printing parameters and we are considered in that initial charging rated charge capacity we are given. So this is that electrode fabrications details for materials and fabrication method. This electrochemical characterization studies we are given in that electrochemical workstation. We run this full charge and discharge cycle for potential. CV studies at different standard and galvanostatic charge and discharge profile at different current density. Then finally, we're talking about that so far that using the 3D printing in that green biotechnology, what way application we are used. But this compared to the traditional method, we are using the 3D printing advantages is freedom of design, complex structure we can easily make, reducing that fabrication cost is very cost effectively a novel design. But in challenges and future scope, so still that material is limitation is there, scaling issues is there, the cost of the material we are still focusing on. In future scope, AI should be integration and smart material. So still, when we're talking about the smart material, 3D printing, and then 3D printing is evolution into the 4D printing, 5D printing, and 6D printing. So when we're talking about that 4D printing, so like what we did here, discussed about the 3D printing, the material, that material having that shape memory alloy, that is the smart material. If we are using the smart material to printing that same material, that is the 4D printing technology. So these are the future scope of the research is still open and decentralized system. These are the references. And this is my team in Genovamic, and this is collaborated with Pondicherry universities. This is the applications. Thank you.
Thank you, sir, for a great presentation. So always 3D printing is also a very interesting topic for research. And few questions were asked by the students like what are the key challenges in developing some stable or functional bioinks containing some living cells or enzymes?
Living cells.
Yes.
Can you repeat the question?
Like, what are the key challenges will be faced developing the stable and functional bioinks containing the living cells or enzymes or
Yeah. So the basic stable attain is when we're talking about that loading material into the syringe. This is all that right now I showed is stable catalyst material. So when loading into the syringe with the pressure, when we extrude the material, this material, it is fine. But when we're talking about the living cell material for bio ink, for living organ tissue printing, when we are loading that, so that where that material stability will attain, no, the pressure we have to consider. If we're giving high pressure, the material high chances to death. So we have to optimize that optimal pressure range to extrude the material. So this is the condition, and then that system of that machine enclosure box should be fully sterilized and to print it as the standard conditions. How we are optimizing. Then ink basically optimization is lots of trial and error basis only we are attaining. Once we are printing the scaffold, then we are basically checking that normal standard procedure, how that material is forming, that post-curing method, everything is done only. We are considering about that strength of the scaffold, this all we will attain. When we asking about that bio ink material, so for basically bio ink material, we working on for bone material. So for bone material, that it's a hydroxyapatite ceramic material will be taken, and then like for medicinal property, we will add it amexin. So that material like the same technique, but we have to use that suitable binder with that limitation, increase the performance. We optimizing and printing, post curing and post process treatment. We will check. But when we coming into that living organs printing, that living organs printing, that flow we have to consider is motor driven flow. We have to play with that.
Okay. Thank you so much for your presentation today. Thank you for accepting our invite for the day. And now I like to invite Dr. Pradeep Kumar, Honorary Secretary, Coimbatore Local Center EA, to formally propose the vote of thanks.
Madam, am I audible? Madam?
Yes, sir. Sir, Pradeep, sir.
Madam, am I audible?
Pradeep, sir. I hope his
Madam,
connection
am I
is
audible?
on. Yes, sir. You are audible, sir.
Yes, madam. Thank you. So, good evening to one and all. Gives me immense pleasure to propose the formal vote of thanks for this IA technical webinar on green biotechnology for environmental sustainability under the aegis of Chemical Engineering Division Board, IEI, organized by Institution of Engineers India, Coimbatore Local Center, in association with the Division of Biotechnology, Karunya Institute of Technology and Sciences, Coimbatore. So at the outset, I extend my sincere gratitude to the President of IEI, Chairman of Chemical Engineering Board, members of Chemical Engineering Division Board, Council members, and Mr. Saurav Dasgupta, Executive, IEI Headquarters, for their constant and continuous support for all our activities. On behalf of IEI Coimbatore Local Center, I extend my felt thanks to today's esteemed panelist, Dr. Vivek Suresh Dalvi, Program Manager, IIT Bombay, Research Hub for Green Energy and Sustainability, Bombay, Dr. K.K. Prashant, Director and CEO, Symbion Labs and Scientific Services Private Limited, Chennai, Dr. T.S. Maniganda Maharaj, Co-Founder and Director, R&D, Xenobiomic Research and Technological Development Private Limited, Puducherry, so for accepting our invitation to address this program. My sincere appreciation to the Dean Engineering and Management of Karunya Institute of Technology and Sciences for continuously interacting with our IIA Coimbatore Local Center and for the dedicated support in arranging various technical activities. I thank Dr. M. Sendil Kumar, our beloved chairman, IIA Coimbatore Local Center, for his welcome address and guiding all our center activities. My special appreciation is due to Dr. A.K. Priya, Committee Member, IIA Coimbatore Local Center, and Professor, Division of Biotechnology, Karunya Institute of Technology and Sciences, for her efforts in organizing this program. Finally, I extend my sincere thanks to all the participants of this webinar, IIA Council members, committee members, IIA corporate members, industry members, faculty and students from various institutions for their participation. Once again, on behalf of Institution of Engineers India Coimbatore Local Center, I thank you all. Thank you, ma'am.
Thank you, sir. We formally close the webinar.
Thank you, sir, ma'am. Thank you, sir. One last announcement for the participant. All participants are requested to register themselves and pay a token fee of rupees 300 plus GST for obtaining the webinar participation certificate. Thank you. Thank you all.